I chose this title when Bio met Nano as sort of like a hint to the film When Harry Met Sally, when Harry and Sally sort of met over different encounters over different years and finally sort of come to fruition as a couple at the end of it. But I'm going to take you on a similar journey where random events have come together and I've met with various people and collaborated with them, and as a result, sort of led to some, well, what I think personally is some cool research myself. But I would say my life in academia is about taking opportunities. I think it was Jim Murray that said to me that I'm very opportunistic in my research areas. In other words, if I see something that I think is quite interesting, I can benefit from it, or I can contribute to it, I will sort of try and get into that area. And that means I do a lot of little things outside of the core research. So I've even got involved in SARS-CoV-2 and COVID by looking at various molecular aspects of how the virus works, including things like its exonuclease. We even looked at glycosylation on a commercially important enzyme called horseradish peroxidase. We've worked on various other enzyme sisters, everything from subtle lysine serine proteases to esterases. Commonality between those two is that they say have the same caloric mechanism. We've done a bit of protein engineering as well, which I would still argue is my core as a group's core activity. Some work with Jim Murray on luciferase engineering, and some work with other collaborators outside the university on designing sort of brand new cofactors to be incorporated into proteins. We've even done some technology development in the area of protein engineering as well, coming up with different ways to create new protein scaffolds, to my very first paper here at Cardiff, where only myself as the author on it, because back then I was in the lab. I did do some work in the lab productively, and I was the only bod around in the first couple of months when I started working at Cardiff, so I thought, well, I might as well get started and do some actual research. And that was my first paper to come out of that. But proteins are at the heart of my research. And there are there's about 3 or 4 proteins that throughout the last so many years at Cardiff that have come sort of embedded in the idea of the nano-related work. First one here is cytochrome B562. It's a very small helical bundle protein which binds heme in the middle, and it's thought to be involved in electron transfer. Another one, which maybe the students in particular get familiar with in my final year lectures, is green fluorescent protein, which is, as all of you know, is a ubiquitous marker for use in cell biology. But it has a lot of other funky uses as well, which we'll talk about in a second. There's a beta-lactamase. Now, beta-lactamases are a very important class of enzyme because they're one of the main instigators of antibiotic resistance, especially to things like the ampicillin-type antibiotics. And essentially what it does is cleave the essential bond here, the amide bond here, which actually is the active bond in terms of how it inhibits the transpeptidases. But because ampicillin and the penicillin-type antibiotics are natural products, and these beta-lactamases were naturally present in the background before we started using them, nature also came up with a natural inhibitor of these beta-lactamases, and that's called beta-lactamase binding protein or an inhibitor protein. And what that does actually, it binds a lot of different of these beta-lactamases and forms a very tight interaction with them. So it acts as a natural inhibitor of these beta-lactamases. And I guess this all relates through to the key things that I'm interested in, is how sequence translates to structure and ultimately function. But more interestingly is how new sequences that we can engineer and we can generate can lead to new structures and function. This is the design and engineering part. So nanotechnology and biology. Well, if you look at these things in terms of their absolute size scales, they work pretty much on exactly the same scales. Nature at the molecular level is the same as where nanomaterials on the sub 1 micron size ultimately between, say, 1 to 100 nanometers. And they also work at the molecular level. All those molecules inside your cell, they are working as individual molecules. You might see a general phenotype coming out from that, but ultimately it's all how those molecules are working and how each of those molecules work. And that's the same as what you want to work at nanomaterial level. You want to see how individual molecular systems are working. And that gives rise to generating, if these two systems are compatible on the size scale, can we bring those two together, biology and nanomaterials and nanotech, to create these what we call bionanohybrids? But also, if we look at nature, nature also is an inspiration for a lot of materials, whether macro materials or nanoscale materials. So can we use biomolecules to inspire and to be integrated with these nanoscale systems in terms of the chemistry, physics, and ultimately how we engineer these systems to be built? So we think about a nanomaterial, something like a carbon nanotube. Now, a carbon nanotube is just a cylinder of carbon atoms all linked together in this hexagon arrangement. But they have very useful intrinsic properties. Graphene always gets everybody interested because that's what won the Nobel Prize. But it's an argument that all a carbon nanotube is a wrapped up form of graphene. Now, they're great materials in themselves because they have very good electrical properties, very good mechanical properties, and optical and thermal properties as well. And the ultimate thing is, all this is one big cloud of electrons that's spread out across this entire structure. So they're very sensitive to local changes, especially in charge. If something comes close to that electron cloud, it can perturb that electron cloud, therefore change its properties. And a typical carbon nanotube can be anything between 1 to 2 to 3 nanometers in its width. Now, a protein can be about the similar size scale as that, but that ultimately has very useful function. It has a diverse range of functions, everything from molecular recognition, being able to identify and bind other things, catalysis, being able to turn things over, and especially in terms of GFP, what we call charge transfer. Now, that becomes quite important because a lot of devices that are so important to our everyday life involve some sort of charge transfer process. So if we think about just having our carbon nanotube on its own and it acting as sort of like an electronic component, well, that's fine. Electrons can flow through it. It can have metallic or sort of conducting properties, or it can have semi-conducting properties, like part of what a modern electronic component is. But ultimately, unless you put something in there, nothing changes to that overall conductance profile. Functionalization is therefore the key. You need to put new properties into that carbon nanotube to give it something useful, to make it change its signal, in other words. And that's where integrating a biological molecule becomes quite important. So we bring our protein into the system, we can potentially get communication between the two systems. So now we have an input coming in. If we input sort of a biologically related input related to the particular molecule we're looking at, that can have an influence on the conductance properties going through the carbon nanotube and thus generate a change in signal. That's where the biomolecule becomes quite useful. It acts as a modulator of the function of the basic nanomaterial. And that's important because it can form the basis of biosensors, responsive materials, including molecular electronics, and even open up opportunities for fundamental single molecule protein studies. The challenges is that integration of the two. So we need to access and thus understand how individual biomolecules work. So this is green fluorescent proteins spinning around there. We have an ensemble of average of millions of molecules, and that's what most people will see in a spectrophotometer or fluorimeter. They will see just that signal and think, that's great, I see that signal, it's wonderful. But that is an ensemble. That is contributed by millions and billions of molecules. What's actually happening is those individual molecules are doing something very different to the ensemble. Fluorescent proteins, for example, will blink on and off. Individual molecules will go through different states. And this is actually the difference between a single mutation in a fluorescent protein, which Dave, sitting in the front of the audience there, generated this image for us quite recently. Thank you very much. So if we want to enhance those single molecule properties, because they're going to be very different to the bulk, we need to know how those two components, how the biological molecule, the protein, is going to interact with a nanomaterial such as the carbon nanotube. Because if we just did what we call standard labeling and just dumped the protein on there, there's all sorts of different orientations that could be sampled. And we don't actually know, because potentially every single one of these orientations are given a different effect onto the carbon nanotube. So we actually don't know which molecule has given rise to the signal. And as a result, that gives us a lot of noise signal. We're getting a lot of non-optimal interactions. We're getting interactions which don't actually give a productive effect. So it's very noisy, it's very heterogeneous, non-optimal. And if you want to ultimately use this in a device, it's got to have very defined fabrication properties. It's got to be built in a very defined way. So this is where being able to design that interface between the two molecules becomes important. So you get optimal communication between the protein and the carbon nanotube. That's homogeneous, so everything is interacting in the same way. So you start seeing defined signals, and when you put that bio input in, you will see a defined output, change in output and signal from that system. So this is where the story begins. How did I get involved where this bio meets the nano? Well, it actually started when I was a postdoc with Paul Barker in Cambridge. He came up to me and said, let's make this conducting wire of proteins. This is cytochrome B562, going back to those. That's probably the original protein that started it. He said, let's build this funky protein which doesn't exist in nature, which can bind DNA and potentially be templated on DNA and form a matrix of these proteins. And the key thing was that this cytochrome B562, which is shown in this helical bundle there, has this heme in there, and this heme has the iron in the middle, and that can undergo, that's got two different redox states, Fe2 plus and Fe3 plus. Now that's quite incredible because it switches between the two. Now that is an electron transfer process, which becomes important in a second. So Paul was the original inspiration for this. I left Cambridge, but we didn't get to work on that DNA aspect, but quite a few years later, we actually were able to do this using synthetic biology to what we call link protein to DNA in very defined systems. We use something called click chemistry, which I'll go talk about in a second. And we were able to make these protein DNA hybrids. Now the great thing is we could put different DNA sequences on different proteins. And that's quite cool because we can also build what we call a factory floor out of DNA. We can create a tile of DNA molecules, which is shown here. And this was done with Eugen Stoltz. Okay, chance meeting up in Leeds. We just met, we got chatting, and this is what came from that meeting. So he could build these tiles and stick up these little bits of DNA at defined positions on that tile. And what they act as, these two bits of DNA, one on the protein, one on the tile, they bind complementary to each other. So we can now assemble a system with proteins embedded. We can see this is atomic force microscopy. Little blob there is a protein on a tile. They assemble. And funnily enough, when we saw that, the enzymes seem to perform better on the tile. We're still not sure why they perform better. Still don't know to this very day. But when we assemble them on the tile, the enzyme activity went about 3 or 4-fold. So we showed there, but we can generate these bionanohybrids and assemble them in a defined way. But going back to the main story, a key thing is, I'll ask you, do you know what a field effect transistor is? If you don't know what a field effect transistor is, just pick up your phone, look on this. The field effect transistors basically allow modern technology to work. They are the switches. They are what generates your 0 and 1 ultimately at the end of the day. These are so incredible. Moore's law is related to transistors. Transistors are, the more you can get per unit area, the more processing power you get. So it's probably the most important electronic component out there. And the key thing here is you've got a source and drain, you've got the semiconductor material in the middle, and you get conductance through that until you apply a gate to it. In this case, it's a voltage. And that applies an electric field across this and changes the conductance properties. And therefore it changes that conductance property, so it gives you that switching effect. So the gate is really quite important. Can we replace that gate with a biomolecule? So in other words, the biomolecule controls conductance through the system. And that's when, a couple of years after I arrived in Cardiff, I met with two colleagues from physics, Emma MacDonald and Martin Elliott. And we looked at, I was working with cytochrome B562 at the time. We thought, wouldn't it be cool to measure single molecule conductance, the conductance through an individual single molecule? And so that's what we looked at. We did the protein engineering, and Emma and Martin did a lot of the sort of measurement. And to do this, we had to engineer the cytochrome, because we needed to get it bind to electrodes, which were gold. And these electrodes, the thiol group here, these cysteines, very good at binding gold, very strong bond with gold. And because we can put those cysteines at wherever we want to in the protein, we can also change the orientation. So we actually generated a molecule with two cysteines there. We actually generated this one here with a cysteine at a different place. So we could look, not only just does this conduct, but how does orientation of the protein change that conductance property? So quite a simple, engineering-wise, quite simple, but very effective to look at mechanisms of conductance. And the interesting thing we saw, first of all, the one thing we saw, proteins are pretty damn good conductors, actually. They're better than some what we call organic electronic conductors. And secondly, we can use that heme bit in the middle there as well to gate the voltage, gate the conductance. So these are individual protein molecules here on a gold surface. And the brighter they get, the more conducting they are. And you can see at these low voltages here, it's very weak. Get up to this what we call the 0 point, it's conducting, and that is low conducting at the more positive voltages. But that's related to that central heme ion. Because what happens is, at these low voltages we apply to the system, it's in one redox state. At the high system, it's another redox state. When we come close to 0, we're at that equilibrium between the two. So now that's the perfect point to get electron transfer for electrons to come into the system and to come out of the system. And this particular curved property we see there is equivalent to transistor. So we're now using the protein as a gate. And this is the first idea of a protein-based transistor. And that's 5 nanometers in length. So I'm going to digress a little bit. And another chance encounter I had was with Eric Tipman, who came over from the States to work in Cardiff for a while. And he introduced me to something called phenyl azide chemistry. Now, phenyl azide chemistry is quite important because it can do lots of useful things. But we need to get it into the protein first. And the way we get it into the protein is to expand the genetic code. So expanding the genetic code means we can put amino acids into a protein which are not normally there in the natural repertoire. Because those new amino acids, no matter chemistry we require, properties we want, and because we can incorporate them recombinantly, we don't have to chemically synthesize it, we can get lots of the protein as well. The result is we get designed incorporation. We get to choose where that non-natural amino acid goes. It doesn't just randomly get incorporated into the protein. We define where it goes. So if we use that in terms of nanotechnology and assembly systems, we get precision modification, which is what we need. And we get high specificity. Because that chemistry is not there in nature, any reaction it undergoes will be unique to it. So we can bring new chemistry that we couldn't normally sample. And that allows us then to look beyond biology in terms of what chemistry we can do and what other molecules we can get it to interact with. So it's quite a straightforward process, actually. I say this, but it is fairly straightforward. You take a gene of interest, you decide which residue you want to mutate, you do the site-directed mutagenesis, you put a stop codon there. You put a particular stop codon or the amber stop codon. Now that's the codon that becomes reprogrammed because we put in a plasmid like this. This contains a new amino acid cell tRNA synthase and a new tRNA which recognizes that stop codon, recognizes non-natural amino acid, and where that stop codon is, you now get incorporation of the non-natural amino acid. So that now allows you to put a hell of a lot of chemistry in there. If you want an easily accessible review to look at that, Athena Zitti's recent publication, The Biochemist, is a nice one to look at. Good introduction to it. The result is the non-natural amino acid actually becomes itself a chemically genetic switch, in that without the non-natural amino acid, you don't get any protein produced. With the non-natural present, you then get your protein produced. But why is phenylazide chemistry so useful? Well, we have the photochemistry. Photochemistry is that this particular chemistry here, when you shine light on it, it loses molecular nitrogen and becomes a reactive nitrogen radical, which likes interacting with electrons. Carbon nanotubes have lots of electrons, and therefore it will assemble. This is a picture of individual protein molecules in AFM graphene. It will assemble in a very defined and site-specific way. You also get a direct covalent attachment of the protein into the carbon nanotube. So you get an intimate interaction between the protein and the pi electron system. So we looked at this. We looked at two different residues with two different positions with respect to the chromophore in GFP. We could assemble those quite nicely, adding UV light to them. There's a single individual carbon nanotubes. And then the next stage is to put it into devices. Now these devices have one single carbon nanotube spanning across two electrodes. So these are quite small things. So along with Ollie Costell over in pharmacy and people like Dave as well in the audience, we looked at what these did at the single molecule level. And lo and behold, we attached these proteins to carbon nanotubes and it actually changed their properties quite significantly. In fact, quite a lot. So it wasn't as just putting the protein on the carbon nanotube changed fluorescence slightly. It changed it quite a lot, so it suggested there was communication between the two. The two were talking to each other. And what we think is it was charge transfer happening between them, because what it was doing was when GFP and a lot of things, chromophores are excited, they can release charge from the system. I guess the classic is photosynthesis with a light harvesting reaction, yeah? And it might be doing that with a charge is circling amongst the carbon atom and maybe coming back to the protein. So therefore, these are a lot dimmer, but they stay on for a lot longer. So there is a definite communication between the two. So we thought, well, okay. So another chance encounter via email this time with Ivan. And he wanted us just to attach some of our GFPs to his electrodes, which I showed earlier on, see what they would do. When we shine light on it, what would happen? So we took the one system, which is what we call AZV132. So it's at the one end of the barrel of the protein, quite far from the chromophore. We shined light on it, it changed the current, but it didn't recover. It just stayed down. Then they applied what we call a reset voltage. It went back up to its original level. Applied light, went down, didn't switch back. Then switched up again, so we could reset it. So this actually occurred over quite a numerous cycles. And actually, this data was taken 6 months after we constructed those electrodes. It's not as if we did it straight away, and then these are 6 months, and actually went through a dry-in cycle as well, and were reconstituted. This is memory. This is how computer memory works. Yeah? This is optical memory. So we're now using GFP to change states, the conductance, and then reset them when we want it to reset. When we took it at the next point, where the attachment position was close, put the chromophore close to the carbon nanotube, we put light on, change the conductance properties, turn the light off, they'd recover. Switch the light on, conductance property changed, etc. through that cyclic process. So we're now controlling, this is what a transistor is, yeah? You put in the light on, changing conductance. So now GFP is acting like a molecular electronic component. It can be translated to nanoelectronics. So the other aspect is biofets, what we call field effect transistors. We've already talked about that. Biofets is where we replace that gate voltage by a biological event. So the biological event gates that conductance. This can be the basis of nanoscale biosensors, where you can construct these devices, where you have a target or something you want to detect, a receptor, something with the molecular recognition properties to bind the target, And that receptor gets incorporated into a device setup called the transducing setup. And that means that anything that binds here changes the conductance through, and as a result, you get a change in conductance output, which is a result of detecting your original input. And what we'll look at mostly is how local charge affects the carbon nanotube. So changes in how the charge itself, close to the carbon nanotube, changes the inherent sort of the conductance properties of the carbon nanotube itself. So Emir, who I was already collaborating with, went away to a conference somewhere. He met a guy called Matteo Palmer. And Emma, he's a very generous individual, said you should talk with Matteo because he's doing these sort of device sort of setups. Why don't you have a chat with him and see what you can get going? So we started that, and one of the first things we started doing was looking at biosensing. And this what we did is we put this molecule called pyrene. Now pyrene is essentially a small part of a graphene molecule. So it stacks very nicely and forms a very strong bond with the carbon nanotube via a process called pi-pi stacking. And we stuck this bit of chemistry on the end of that pyrene. Because that bit of chemistry, when you add it to this bit of chemistry, the phenylazide chemistry, especially the azide bit, will form a very defined crosslink. And that's what we call click chemistry. Now, people like Caroline Bertozzi won the Nobel Prize for this just a few years ago for its use in biological systems. We're using it here in sort of like a biochemical hybrid system. But through that, we can get very defined attachment and assembly of these systems. So we can now get a protein attached in a way we think we can get in terms of how it interacts with the carbon nanotube. So how would we go about building these? Well, actually, we start with the basic carbon nanotube. And then this is the size of the devices. These are the actual devices we're looking at. The electrode gap's about 300 nanometers. We add down our coupling agent, which is the pyrene, that cool bit of chemistry we can attach our protein to. And then we come in, we just then attach the receptor, wash it away, so we only have our single defined setup there. And what we looked at this for originally is going back to my list of those 4 proteins. Now we're back to those last 2 proteins, the beta-lactamase inhibitory protein BLIP and the beta-lactamases. We now decided we could use this system, especially BLIP. Now BLIP will bind a lot of different beta-lactamases. It's not just one BLIP to one beta-lactamase. The BLIPs will bind basically any what we call the class A serine proteases, and that is a lot of serine beta-lactamases, and that is a lot of serine beta-lactamases. One would argue they are the predominant form out there that cause resistance to penicillin type antibiotics. And what we want to detect is a beta-lactamase, because these beta-lactamases are what do the damage. That's what gives rise to resistance to penicillin type antibiotics. And the cool thing is they form a very defined, strong complex between the two. And we have structure of these complexes as well. And this is the key now. So our BLIP can act as what we call the receptor, the thing that undergoes molecular recognition in this nanocarbon setup. And the cool thing is these beta-lactamases, we all look at them in terms of these cartoon diagrams or particular chemistry. They have a surface. That surface is very electrostatically diverse, but has very defined electrostatics. Every protein will have its unique electrostatic surface signature. And that's the key thing, because that electrostatic surface is what does the gating. That's what, when it comes close to the carbon nanotube, will change the nature of the electron cloud and the charge carriers in that system, and as a result, change conductance. The key thing, though, is getting this right. There's something called the Debye distance, which is quite critical. If you're too far away, you don't get that transfer of the electrostatic down to the carbon nanotube. So it's good, you need to get that close. If you attach it wrongly as well, you might get blockage of it. So if we did a random attachment, we get so many different orientations of BLIP on that system. You might only have 1 in 10, which is capable of generating a signal output. So as a result of that, you've only got 10% generating something useful. Ideally what you want is everything working in the same way. So anytime there's a molecular binding event, you get a change in signal. That's where you get a higher sensitivity then. And that's where defined attachment plays a big role. And through defined attachment, because we can use that non-natural amino acid chemistry, we can put it in a different position in the protein and sample a different surface of the incoming beta-lactamase. And that should result in a different electrostatic change. So not only are we just restricted to one place, we can actually we could potentially shift it at different positions and map the electrostatic surface of an incoming analyte and use that as a signal. So what we did is we chose quite a few positions. We put the phenolase at different positions. And what we got is these are the two sort of main ones we focused on. We got this one here, 41 and 213. So that's the different residue positions in blip we did the attachment. On this you can see here, the different attachment positions will potentially present different electrostatic surfaces. And what we found is they would regenerate different conductance profiles. So when we added it in this case here, at 10, this one, it presented a higher negative charge density close to the carbon nanotube, which was suggested, which will increase the charge carrier through the system, so therefore higher conductance. In this case, when we put it at 213, it's now presenting a more positive charge density close to the carbon nanotube. So it actually, in this case, decreases conductance through the system. So you can see by altering this, it's the same analyte we're detecting, but it's giving very unique, distinct signals. So we've got two approaches now for linking proteins through to the carbon nanotube, photochemistry and click chemistry. Now, the click chemistry is all very nice and it's all very easy, just put something down. But the problem is there's this big linker in between. And that's quite flexible, but it also creates a gap. You might think, oh, it's only about 1 nanometer gap, but that could be enough to shift it beyond a detection range. So it's very difficult to discriminate what's actually happening with that compared to the direct interaction where you're basically putting the protein right next to the carbon nanotube. So while we've already discussed the issues with non-specific attachment, we can also do specific attachment and actually not do it very well. In other words, the designs we put in could be crap. You could get steric blocking, beyond detection range. You don't deliberately do this, of course. That's not what you aim to do with these designs. It's just when you put them into practice, that's what might happen. So ideally what you want is to be able to design things more accurately. So when you build it, it will have those particular characteristics you want. So it gives you an idea of what to expect. And that's the idea of predict before you build. And that's where another chance encounter with an individual from Montreal called Sébastien Cote, he sort of helped us solve that problems. Now, the idea is that molecular dynamics becomes very important in this process. I would consider that the next development in protein design. And Georgina Menzies, also in the audience, has been a key mover for my group especially in being able to undertake molecular dynamics. I think it was coming in during COVID and a lot of the group moved to molecular dynamics. And Georgina was pivotal in helping move that work forward. And the key thing is, when we design proteins, they actually move. They also use water as well, that pesky thing, water, that proteins work in. And they're also modified. So when you look at a basic protein design, you look at a static model and how that thinks, that is probably just looking at one snapshot of the whole process. If you look at this particularly, all these are critical to function, all these residues, and that water is critical for function. See how they're moving? That's over 10 nanoseconds as well. This becomes even more pertinent when you bind it to what we call a static object, because there will be free rotation around the bond you attach it to. And this is the intimate contact. And these are all the sort of sample trajectories that this blip attached to this position can sample. So things are moving around. And we need to know the population distribution in this, how much at any point in time a particular, it's in one configuration, and what charge is important on the carbon nanotube. So it becomes very important we understand these molecular displacements and how they change the interaction with the carbon nanotube. So we looked at this again. We went back to our two positions at 41 and 213. And we looked at just molecular dynamics of that, just to see what was happening at 213 and 411. We found that Becky Gwyther did this work. She found that they sampled different rotamers, so different sort of configurations of the phenylase A backbone, and that's free in solution. When we sort of docked that basically onto the carbon nanotube, we found that for this one, this R1, which is the majority one it sampled, would create steric clashes, and R3 would here as well. So we ruled those out. But, you know, we thought at least these ones would be able to form sensible, feasible attachments. We're thinking, well, that's not very good. It's all the way over there. This one looks pretty reasonable, but it's actually the least sampled form. So we thought, fair enough. But we need to look at this when it's actually bound to the carbon nanotube. So they went away with Sebastian, did that, and we come up with these whole ensemble of structures that come out from that. And from that, we can then, when we map the analyte and bind the analyte to it, we can come up with density electrostatic maps of what interaction is working with in terms of the protein and carbon nanotube. So this is just with a blip protein bound. And you can see that's electrostatically quite distinct. A lot of negative charges on the system there, especially in that hole in the center. And that maps to give a net negative charge on blip close to the CNT surface. And we look at that experimentally when we just put the blip on there, we see that change. These are what we call P-type semiconducting carbon nanotubes. Don't worry about that, but when negative things come close to it, it should increase conductance. And that's what we're seeing here. So our predicted matched this in a very sort of protein onto carbon nanotube system. So it's not about the overall charge, it's about what's close to the surface. So now we want to see this with the analyte. So we now took two beta-lactamases. One was the classical 10-1, and the other one was a KPC2 that we took. Now they have very distinct different electrolytic profiles. So when we did the modeling of that, we looked at charge distribution over time and looked at what was happening. When we looked at 41, it was having very little effect, very little charge effect on the carbon nanotube. But when we looked at the 213 position, that looked like not only were we getting distinct charge effects that would transmit through to the carbon nanotube, the 10-1 and KPC were very different. 10-1 would put a negative charge close by, predominantly, whereas KPC would put a positive charge close by. And it's binding to exactly the same target receptor protein and exactly the same position. So we predicted that beta-lactamase binding will have a small effect when it's in this form, but this one will have a larger effect with 10-1 and this KPC2 imparting different electrostatic profiles on the carbon nanotube and therefore different conductance. We said that 10-1 would increase current, KPC would decrease current. So those are our predictions. When we put that into the system, we got exactly what we wanted to see. Or what we observed was BL binding. It did generate a small increase, with TEM slightly bigger than KPC, but we predicted CHEM would impart a slightly higher. But when we looked at 10-1 and KPC, 10-1 gave a much bigger change in conductance. So it increased conductance like we thought it would, and KPC reduced conductance like we thought it would. So we're now seeing, and we also get the concentration dependence on that. So we're seeing what we wanted to see there. We're seeing those changes. So we've now got a way to try and predict what's happening. And because each beta-lactamase, even some of the single point mutations, quite common in beta-lactamase mutations, are they will change one charge for another charge, or change something from having no charge to a charge. So it will change charge on those key mutations which give rise to changes in activity. So maybe we could look at that as a sense of detecting different types of active beta-lactamases. This idea of multiplexing. We could potentially create an array of different blips attached to different positions, so therefore we could build up a sort of knowledge upon how a beta-lactamase would bind, what signal it would generate, therefore what beta-lactamase it is. In conclusion, I think that engineering proteins is important, not only for general sort of basic understanding of proteins, but it can facilitate their use in new areas, especially with the use of phenylase A chemistry. This new chemistry we can incorporate through an expanded genetic code gives us this power now to do things we couldn't previously do with proteins. But the options with phenol are the click chemistry and even photochemical attachment. And now there's this move towards modeling and design, and we're no different to that. The Nobel Prize for Chemistry this year was for protein structural modeling and design. And this is probably the next step forward, is being able to bring dynamics into that aspect and being able to predict these systems before you actually build it. And are proteins the future of electronics, not just as biosensors, but maybe the individual components in your, maybe you will be powered by GFP, your processes might be powered by cytochrome B562 in the future. So I'd like to say thank you to all the people who have contributed towards this work. So Becky Gwyther did a lot on the biosensor work, and with Ben also with the blip and 10- and the beta-lactamase work. Georgina for all the help with the molecular dynamics. Ollie and Dave have done a lot of the single molecule work, even beyond this current project as well. And Martin and Emma, together with people in their lab who were great collaborators at the time. And Ivan, Eugen, and one of his postdoc, Gabby, who did the work on the DNA assembly. Matteo and people from his group who helped us do the measurements on the carbon nanotubes, and Sebastian and Will and Kevin, who are part of a fledgling and ongoing collaboration. And these funders as well for all their money that they give us. And just a quick note that Sam Winter was a student in Cardiff. He was one of my former personal tutees. He was a good friend as well. We did some collaboration work with him. His name's on 2 papers that we did on various things when he was a PhD student over in Bath, and he sadly passed away last year, way too young. So this is in memory of Dr. Sam Winter. Thank you very much.